Hello, everyone. Welcome to the Sernova Corp presentation. My name is Dr. Philip Toleikis, President and CEO of Sernova Corp, and I just want to show my appreciation for being invited to the Cell and Gene meeting on the Mesa in 2020. Today, I'm going to go over Sernova Corp and in terms of our regenerative medicine approach and update everyone on our clinical trials as we're moving forward. So Sernova itself is an innovator and a leader in this field. We've been working on the cell therapy approach for therapeutics for over 11 years. And we are now in clinical trials, and I'm going to show you some of the successes that we've had with our therapeutic cell and device approach. We are working both on a diabetes program, and we also have a Hemophilia A and Thyroid preclinical programs for which we have validated our technology in. Sernova's approach takes three types of applications here. So first, we've developed what we call the cell pouch, and the cell pouch technology is highly scalable. And this is an implantable device that is about the size of a business card that fits deep under the skin. And the approach that we're taking is that the cell pouch itself is used to create the organ-like environment for the therapeutic cells. Our therapeutic cells we start off with human donor cells for our clinical programs because we want to be able to see how the real cells in your body work in our device. And then we also have stem cell derived programs to provide an unlimited supply of cells. And these cells release missing proteins or hormones into the bloodstream appropriately. From the immune protection part of our technology, we have a number of different approaches, and we're working on both a polymer approach to be able to protect the cells locally within our cell pouch chambers, and also we have a gene editing approach, which is one of the latest approaches that is being taken currently. From a patent portfolio, Sernova has very, very broad patent coverage, both on medical devices for delivery and cell transplantation, for therapeutic cells, as well as our local immune protection technologies. And our patents, and we have full patent claims and multiple divisionals in virtually every country in the world at this point. Regarding our pipeline, our primary clinical program is for type 1 diabetes. And here we are currently working on a phase 1/2 study at the University of Chicago. And we're using human donor islets, again, which are the real thing that are found in your pancreas within our device. And I'm going to talk about the study in a lot more detail. We are also in parallel working on locally immune protected stem cell derived cells to provide an unlimited source of cells for a much, much larger population. We have proof of concept studies in both Hemophilia A as well as for thyroid disease that I will go over. And both of these programs are also moving into the clinic. With respect to our therapeutic cells, not only are we working on human donor cells and stem cell derived technologies for which we have an exclusive worldwide license to, we are also working on collaborations with large pharma companies who have their own stem cell derived programs for diabetes, with the opportunity to be able to have the licensing and marketing distribution to build on our technologies as we're moving forward. From the immune protection approach that we're taking, we were able to use immunosuppression drugs successfully with our therapeutic cells, and we've shown that this works very well in the clinic. We have stayed away from immune protected macro devices because we believe the combination of immune protection as well as trying to get vascularization is really an oxymoron, and we've seen some failures in the field in this area. Instead, we've been working on protecting the cells from the immune system within the device chambers itself, and thereby working on a conformal coating technology, which we own and have licensed applications to. In addition, as I mentioned previously, we are also working on a gene editing approach whereby we can take the HLA gene and place that into the starting stem cell for our stem cell-derived technologies. So we're using a number of different technologies to be able to protect cells from immune system attack. Now, if we move on, we have worked really hard on having a biologically compatible delivery device. And we've looked very closely at how the human body works, and we've programmed our device and cell technologies to be able to match the needs of the body. So the way we do our applications is a little bit different than everyone else's out there. And we insert our device deep under the skin in a very short, rapid procedure. And then we allow the device to sit under the skin for several weeks while tissue and highly vascularized tissue grows into the pores of the device, forming these beautiful vascularized tissues with pores in them to allow the cells to be able to go into the vascularized tissue chambers. Then we will come in and transplant the cells directly into these highly vascularized tissue chambers. And then rapidly thereafter, there is engraftment, which means that the vascularized tissue then surrounds the therapeutic cells, and thereby allowing those cells to survive. And then in the case of diabetes, be able to read the blood sugar levels in the bloodstream and be able to reach insulin appropriately. And we've validated this technology in multiple ways. So in terms of Sernova's strategic development approach, there are two ways to move forward in this type of technology. One is to try to combine all of the technologies together and go for the home run. And when you go for the home run, you also have a lot of failures along the way. And what Sernova has done instead is taken more of a stepwise approach to achieve success. And that's why we're working with human donor islets at the beginning, and we've been successful in our clinical trials doing that. At the same time, we're able to learn from what we have with our human donor islet program to be able to move into our stem cell-derived technologies. And this has allowed us to have continuous significant success in the field, and we believe we're a leader in the field because of this approach. So if we move on to our clinical applications here, we all know that diabetes is a very serious problem and getting worse and worse, with over 400 million patients affected worldwide. Approximately 10% of these have type 1 diabetes. And this is the population that we're currently focused on. And what we're looking at is a functional cure for everyone suffering from diabetes. And after 100 years of insulin injections with what we believe is no significant improvement to the treatment of these patients, we are working on and achieving and getting closer to a functional cure. So if you look at our diabetes clinical progress success, we have been developing this program for a number of years. We've conducted a first-in-human study up in Canada. And as I mentioned, we're working on a phase 1-2 trial at the University of Chicago, for which we have some initial successful data that I'm going to be discussing. So if we look a little more closely into the patients that we're focusing on, these patients have severe hypoglycemia unawareness, and this is the most critical unmet need of diabetic patients. It affects about 15% of patients, and there are approximately 240,000 patients in the US alone that have this issue. And what is hypoglycemia unawareness? The issue with these patients is that every time they take an insulin injection, their blood sugar levels can drop to precipitously, and they don't have any awareness that such as the shakes or tremors, that their blood sugar levels have dropped, so they can go into a coma. So I look at it as playing Russian roulette with every single insulin injection. So these people desperately need something to improve them, and a cell therapy approach is the only way to be able to help them. With respect to our US phase 2 clinical design, it's an open label study. Primary endpoint is safety, and we have some key secondary endpoints that actually really matter to the patients that have this disease. The first of these is proving that our endocrine tissue is surviving in our device. And this is really important because a number of other groups have found dead cells and fibrosis in their device. And so we want to absolutely make sure that our cells are surviving. And thereby, we have what we call sentinel devices that we place under the skin with the therapeutic devices. And we remove those sentinel devices at key periods to be able to look at the histology and look at the cells and see how they're doing. We're also looking at a proportion of patients with severe reduction in severe hypoglycemic events, also a reduction in HbA1c, and we're looking at 20 additional endpoint analyses so that we really have a good understanding of how the cell pouch and how our cell technologies are working. So there will be no binary failure in this study. We're really looking at multiple points in terms of how the device and how the therapeutic cells are working in our patients. So in terms of our status, we've received IND allowance by FDA. We've cleared all of the hurdles and we've been treating patients for some time. If I move very quickly through the study design, this is somewhat unusual. What we do is we implant cell pouch, then we do our first islet transplant, and importantly here, this is a dosing study. So we put several doses of islets, and in between, we're able to follow the safety and efficacy of those cells. And then again, at the 90-day time points after each dose, then we remove a sentinel pouch and then we look at how the islets are doing. So we do have some early case study findings I want to go over. And in terms of safety findings so far with respect to our primary endpoint, we've shown no incidents of adverse events. The cell pouch has been well tolerated, it's highly biocompatible. We have no reactions to the device, no fibrosis. And each time that we do our transplant, we've shown that the cells are well incorporated with tissue and vascularized tissue. With respect to some of our efficacy measures and our early findings, one of the first and most important things you have to do in this field is show that you actually are getting insulin into the bloodstream. So insulin is measured by a biomarker called C-peptide, and this is a gold standard for insulin production by cells in the body. So Sernova clinical trial has shown an increase in blood levels of C-peptide, both in patients that are fasting and also following a dose of sugar. So we're getting insulin to the bloodstream. Then, in terms of efficacy measures, we've shown a reduction of hemoglobin A1c that can only occur with cells, not with insulin injections. We've shown a reduction in long-acting insulin use, and we've also shown a 75% reduction in severe hypoglycemic unaware events. In addition, we are working with Medtronic, who are doing continuous glucose monitoring in our study, and we've shown improvement in every glucose monitoring measure, including hypoglycemic unaware events in our tracings from before to after. We have also shown in our sentinel devices abundant, viable, organized islet cells that are surrounded with blood vessels sitting within the collagen matrix, strongly expressing insulin. So this is another level of validation. In our first in human study, we've shown a similar safety in the device as well as the therapeutic cells survival. Moving forward, what our plans are for this year is to finish enrollment of the study, continue to get data, and we're going to be giving regular updates of clinical trial progression as the year progresses. With respect to our Hemophilia A program, I'll be very quickly about this, but we had a 5.6 million euro grant. And what we're doing here is taking a sample of the patient's blood, doing ex vivo gene therapy. And in this program, we've been able to show preclinically that we can correct the factor VIII issues and blood clotting in a preclinical model. And this is moving towards the clinic. With respect to our thyroid disease program, we're working with patients that have benign thyroid disease, and we've been able to do preclinical studies where, again, we've gotten access to human patient tissue. We've removed the tissue, placed it into the device, and again, we've shown survival of the cells within the device and also function of the hormones in the bloodstream. And in this particular application, there are over 150,000 thyroidectomies produced in the US alone. So it is a multi-billion-dollar market. This just shows some of the slides of the tissue within the cell pouch. In terms of our 2020 action plan, Again, we're working and developing and moving forward our clinical trial to full enrollment. We're also moving our immune protection technologies for advanced therapeutic cells. And then we're also looking at completion and driving forward our Hemophilia A program and our Thyroid program. And this is the conclusion of the presentation. And again, thank you very much for the opportunity to present today.